Hello, and welcome to this presentation on non-spherical hydrogel particles for 3D printed composite tissue scaffolds. My name is Gareth Flowers. I'm originally from Powell, Wyoming, and I'm a junior in chemical engineering. In this presentation, we're going to talk about what tissue engineering is, what hydrogels are, and how hydrogels can be used in tissue engineering. Tissue engineering is a multidisciplinary field and is the integration of cell biology and material science. Tissue engineering is used to replicate natural tissues found everywhere in the body, such as nervous tissue, muscle tissue, epithelial tissue, as well as connective tissue. This has applications in cell delivery and cellular regenerative therapies. One of the main tools that we use in tissue engineering are hydrogels. Hydrogels are rigid 3D polymeric structures. They are biocompatible and their high water content makes them very similar to human tissue. They have applications in cell delivery, cell encapsulation, tissue scaffolds, and drug delivery. Here you can see a prepolymer powder, a unpolymerized hydrogel droplet, a polymerized hydrogel particle, dehydrated particle, and a seed here for reference. Hydrogels are photoactive and are generally fabricated on the scale of 30 to 1,000 microns. Manufacturing them smaller than this is usually difficult and slow, but miniaturizing them has many benefits. On the left is a diagram showing the polymerization chemistry involved with the formation of hydrogel particles, and on the right are two reactions. So we can see that our photoinitiator reacts with oxygen present in the system as well as with our prepolymer. The rate constant for this oxygen reaction is much higher than the reaction with our prepolymer, meaning that our photoinitiator preferentially reacts with oxygen present in the system. This phenomenon is known as oxygen inhibition. Oxygen inhibition changes the polymerization of hydrogels in several significant ways, but can be exploited to give desired characteristics. Here we have our prepolymer gel. It's bombarded with UV light, and then we see the formation of this polymerized hydrogel core and this unpolymerized shell around it, which can be removed later in downstream processing. On the far right here, we see how increasing UV intensity can diminish the size of that unpolymerized shell and increase the cross-linking density of the hydrogel itself. General process flow for the creation of hydrogel particles. The shear force from the oil phase creates these monodispersed droplets, which are polymerized, collected, and washed downstream. Here's a video showing the formation of hydrogel particles, and on the right, we have a typical microfluidic device. Here's what a device used to create non-spherical droplets looks like. The big difference here being this modified channel geometry at the exposure site. Here are some pictures of some non-spherical hydrogel particles previously made in our lab. The effects of oxygen inhibition can clearly be seen here and here. Oxygen inhibition and UV light manipulation allow us to exercise greater control over the size and shape, the elasticity, the mechanical and interfacial properties, as well as the degradation profile of hydrogels. All of these abilities allow us to encapsulate living cells in hydrogels and maintain their viability, as can be seen here, where we have a composite formulation of both living cells and hydrogels. These types of composites could be groundbreaking in the field of 3D bioprinting. 3D printing used in tissue engineering offers a greater complexity in design and is a relatively simple and time-efficient operation. It also offers us a more personal and individualized approach to regenerative medicine. Nerve regeneration is one specific area of tissue engineering where hydrogels are highly applicable. Previous methods involve squirting a glob of hydrogel solution into a surgical cavity and doing a bulk polymerization, as can be seen in these top figures. This crude process has given way to a more individual and personalized approach, where a scan is taken of the nerve and a specific mold is made to encase that nerve in hydrogel. This process only takes about 3 hours and can be seen here in these bottom figures. I know you're all wondering how hydrogel particles come to play in this because they seem to be everywhere. It turns out that because hydrogels are biocompatible and some are cytocompatible, hydrogel particles are great applicants for creating resins used in 3D bioprinting of complex tissue scaffolds. Here's a picture of our new Cell Ink BioX tissue printer, which can be used to directly print tissue scaffolds. Combining the cell encapsulation and printability of hydrogels would open the door to a greater range of printing resins and bioprinting applications. These applications of hydrogels are where my research will focus in the ensuing months. I would like to thank Dr. Oki and his lab, especially Cassidy Enlow, for her immense support, and all the people who had to sit through my practice presentations. I'd like to thank Embry, especially Dr. Scott Seville and Dr. Annie Bergman, as well as NIH.